All right. So hi, everyone. I'm very excited to be here today. So this is a project that I've been working on with Dr. Justin Silverman, who's in IST. And we've also been working with Dr. Greg Glore, who's at the University of Western Ontario. If you're familiar with ALDEx2 at all, you'll know him as the package creator, maintainer, all of that good stuff. So the whole point of the talk today is to discuss some of the recent changes that we've made to the ALDEx2 software suite. And basically these changes step away from normalizations. And I'll discuss what I mean exactly by normalizations later. But in stepping away from normalizations, we can include more informative models about the scale or the abundance of our taxa when we're analyzing our count data, our OTU or our SV table type data. So now before we get started, you're going to need the newest version of ALDEx2. And this is brand spanking new. Greg just posted it to Bioconductor 2 days ago, which means if you install it on Bioconductor right now, there's a chance you won't get the most recent updates. I'm not sure if you guys are familiar with publishing R packages, but when you push it, it takes some time to build the binaries and all that stuff. So if you just use bioc install, you might not get the right update. So this is actually a little bit incorrect. Your safest bet is to just install it directly off Greg Glore's GitHub. And that will basically make sure that you have all of the new changes. And if you prefer to install it off Bioconductor, you can do that probably in a day or two, and it'll reflect the same version. All right, so I'll give you all a second install. And I also just want to make a quick note that all of the material for the workshop, all of my data, all of my slides, everything you would need can be found on my GitHub. Sterling also shared it with, I think, your working group GitHub. So it's all available online. Please feel free to take a look and use it however you see fit. All right, so is everybody good to go? Okay. Before I get started, I'm just kind of curious, who of you have used ALDEx2 before? All right, so half the room. Okay, just wanted to know how in depth I have to cover some of these things. All right, so let's get started. So for this whole talk, I'm basically going to be considering a really simple problem formulation. And basically we're just considering a really simple study. How does antibiotic administration affect microbes in our gut? So you can imagine we have some samples, some participants, we give half an antibiotic, half not. We just kind of want to see which taxa are changing in the gut in response to this antibiotic. Research question then is very simple, right? Which taxa are changing in absolute abundance after we give an antibiotic? And then the question is basically, how do we analyze this data? All right, so those of you who've seen ALDEx2, DEC2, all of these wonderful methods for differential abundance are probably saying, what's the point of this talk, right? I can just use those. But we're going to argue here that we can do better than those methods. We can move past some of the assumptions that they make and have more robust inference if we include the concept of scale in our models. All right, so first we're going to just have a few slides kind of discussing what we're doing. And so the first thing I want to make abundantly clear is that we're viewing sequence count data, and sequence count data arises as a series of sampling steps, right? What we're truly interested in is the microbes in our gut. But we can't get that. We get a sample of it. We have access to a fecal sample or fecal swabs that get processed in the lab. And then what we get out is this count data set that's representative of that. But basically what we have is a loss of information, right, from that sampling. We have all of these microbes, but we're only getting a tiny fraction of them. You can think of if you're going to a ball pit that kids play at, right? If you put your arms out and try to catch as many balls as you get, it's more of a function of how big your arm span is than how many balls are in the ball pit. And the same problem arises here with our sequence count data, right? It's a product of our technology, not of the total scale or the total number of microbes in the gut. And so the practical implication of this is that it leads to information loss in the data, right? There's a gap between the data, which we're calling Y, and the system, which is your gut, which I'll be calling W. Furthermore, we can basically say that the data really only informs on one part of the system. It informs on the composition of the microbes in the gut. So the data only informs on the relative proportion of the system, not of the actual scale of the system. And throughout this talk, you might see the notation parallel like W parallel up here, and W perp, and basically I just use that to denote compositional quantities versus scale quantities. If that makes sense. So W perp would be like the total size of the microbes in the gut. W parallel is there's 50% of those microbes are this class versus 20 are from here versus 10 are from here type of information. So what is the practical implication of this information loss? Is that there's a gap between the analyses you do and the research question you're asking. So remember, we're going back to our research question. Which microbes change in response to the antibiotic? What we really want to estimate is something like a log-fold change. That's what I'm showing down here on the leftmost box, right? We want an estimate on the scale or on W of which microbes are going up, which microbes are going down. In reality, we're not getting that, right? We're actually doing some analysis that's a function of our sequence count data. It isn't what we want. It's not necessarily answering our question per se. And how well it's answering the question depends, one, on what the question is, and two, what kind of analysis you're actually conducting, right? Are you using ALDEx2? Are you using DESeq2? Each of those are going to make some assumptions to bridge that information gap that we talked about in the last slide. Does this all make sense to everyone so far? Perfect. All right. So let's think about some of the common options for analysis. So the first one would just be use some existing method for differential abundance. We talked about these. They all rely on normalizations. And basically, again, what I mean by the concept of normalizations is that they use some function of your data to answer the question. And in doing so, they make some assumption about the scale of the data through that normalization. Now, the other option is to reframe the question, to use like compositional data analysis type techniques. But then we're basically saying we can't answer some questions rigorously. We're acknowledging that scale doesn't exist, but then again, we can't answer the desired question of do taxa change between conditions. So kind of like, what are we doing here? Let's go home. It means we can't answer a lot of the research questions we might want to. And if we can estimate something, again, we might be estimating like changes in centered log ratio coordinates or some function, some log ratio coordinate, which isn't exactly what we want to get, right? What does that mean? How does that match back to our actual analysis goal? So there's problems with these methods. I think I've already kind of highlighted what they are. For the first option, this disconnect between what the normalization is saying and like what is actually going on in the data can lead to some really bad implications for inference. Namely, you can have some really wonky type 1 and type 2 error rates. And so in this graph up here, we're basically showing that ALDEx2 and DESeq2 can have this problem of unacknowledged bias. They have elevated false discovery rates as you have more sample sizes. And it's basically a product of the normalization that it's making. It assumes with complete certainty some statement about the system scales, and that statement's often wrong. As sample sizes get larger, you're not buffering against uncertainty because, right, statistical techniques usually are going to collapse in uncertainty. So what buffer you did have is gone, and now you get this elevated false discovery rate or elevated type 2 error type thing. That's bad, right? We should want more data leads to more reproducible and robust inference, not the other way around. The problem with option 2 is that we just can't answer the question we want to answer, which leads to a lot of scientific discoveries just not happening. All right, so in my lab, together with Dr. Silverman, we've been working on this new tool called scale-reliant inference. The idea is, how do you do inference on questions that are scale-reliant, which, again, is probably not the most helpful thing. But we're just saying, instead of taking the first option where we rely on normalizations, or taking the second option where we don't do anything, we can do a middle ground between the two. And that middle ground will basically allow us to principally answer questions that depend on scale and scale-reliant questions. And a scale-reliant question is differential abundance, right? We want to know which taxa change in absolute abundance between the conditions. We absolutely need to know how many microbes are in our gut. Correlation analysis also depends on the scale. So there's a lot of common analyses where you need this kind of additional information. So the whole point of this approach is that we fully acknowledge that the data we observe is a measurement of some underlying system W, and that in the data process, we lose information about the total size of W. We also acknowledge that you might want to do analyses that depend on W, right? You might want to answer a question that depends on the absolute abundance of microbes. However, we note that you can decompose W into both a composition and a scale part, and we can use this decomposition to create models that can answer scale-reliant questions in a robust way. So also, just this is like a super high level on the theory. The one thing we develop in this paper is this concept of a scale simulation random variable. And the whole point of this talk is that we're going to show that ALDEx2 is a scale, like can be written as a scale simulation random variable. If you take nothing else from the slide, I'll explain it in a second. Just know that what we're doing is essentially incorporating a scale, a model for scale in our analyses, and in doing so allows us to incorporate some outside information or even just misguard against errors that we may have in scale. And so scale simulation random variable basically just says, again, right, we want to estimate a quantity that depends on both the composition and the absolute abundance of the data, like differential abundance. And the way that we can do it is, one, by drawing a model for the composition from our data, two, then adding in a scale model, and then three, aggregating those both together to get samples of what we want, right? Our quantity that we want that depends on both. So I know that's a lot. Does anybody have any questions right now? Is anybody confused on anything? All right. So then the rest of this talk, we're going to pivot into how can we make ALDEx2 incorporate scale uncertainty? How can we step away from these assumptions of normalizations and into something more principled and robust through a scale model? All right, and actually ALDEx2 is a great model for this because it's relatively easy to break open and understand and incorporate scale into it. All right, so you said about half of you guys are familiar with ALDEx2. It's basically 4 steps. In the first step, we take our input data Y, so like your OTU table, your sequence variants, whatever you have, and you draw samples using Dirichlet resampling from Y. Like Y is basically your weight on the Dirichlet distribution, and you keep drawing these Monte Carlo replicates. This is like a sample from the composition, right? A sample from W parallel. Then after you have those samples, we're going to apply the CLR, which CLR is the centered log ratio coordinates. I'm guessing most of you are probably familiar with it. If you're not, it's just a special transform and compositional data analysis where you take the log and subtract the geometric mean. So it's just a function. If you have a vector, it's a function of the vector where you take the log and subtract the geometric mean of that vector. Does that make sense? And so after you apply the CLR transform, now you have a bunch of samples, CLR transform data. Then ALDEx2 proceeds by testing each of those samples. You want to test if there's a difference, right? If you have some that came pre-antibiotic, some that came post-antibiotic, we can do a t-test, see if our CLR coordinates are different between the two. And then recall that we drew those bunch of those Dirichlet samples. So we have a bunch of replicates here. So we have to aggregate over all the test statistics. We repeat this for all the Dirichlet samples. We have 100 Dirichlet samples, we have 100 test statistics, we aggregate, return the mean, and then based off that mean, we say, is something significant, is something not significant? Does that all make sense to everyone?
What is the purpose of the resampling step?
So it captures a sampling variability in Y, right? It's trying to capture, it goes back to like, if you would take different stool samples, right, you'd get something slightly different every time.
Mm-hmm.
And it's just trying to capture that like sampling variability step that comes from that issue. Okay. So that's what the motivation behind that is.
Okay.
It's pretty common. A lot of methods do that kind of thing. Any other questions? Okay. So I showed you that graph a couple slides ago about like the increasing false discovery rate, right? And so basically that is a symptom of the unacknowledged bias in ALDEx2. And basically what I mean by that is that if we consider that we have this absolute quantity W, we can decompose it into composition W perp or W parallel and the scale W perp, we can basically show that that CLR assumption in step 2 corresponds to the assumption that the mean of your sample is equal to, or the abundance of your sample is equal to the geometric mean of that sample, or function of it, the negative geometric mean. Does that make sense? Most likely no, right? If you're looking at somebody's gut and thinking about the microbes in the gut, would you expect that the number of microbes in the gut is dictated by the geometric mean? So it might make sense in some situations, but in a lot of situations, it won't make sense. And more problematically is ALDEx2 assumes this with complete certainty. It's saying that is what the scale of the system is. There's no ifs, ands, or buts about it. And so that is actually what's causing these elevated false discovery rates. The geometric mean assumption that I showed on the previous slide is wrong here. And it being wrong as sample sizes increase, it just is placing all sorts of false certainty in things that it shouldn't be placing to. All right, so then what can we do about this? So the whole point of the updates of the ALDEx2 package is that we're going to move past that CLR step and replace it with scale. And so instead of considering our geometric mean assumption, what if we just considered errors in it? What if instead of saying the scale is the geometric mean, we say, the geometric mean is not a bad place to start, but I'm not really certain that's going to capture on it. You know, let's throw a little normal error or something like that on. And doing that alone can actually really improve the results that you get, right? You're considering that little bit of uncertainty in the normalization. So we're moving away from it is the CLR to there's probably some error in the geometric mean, which is implied by the CLR. Does that make sense to everyone? Okay. So how do we do that? Let's go back to our ALDEx2 example. The biggest change is now here in the second box. So instead of drawing samples or applying the CLR transform, we can just draw some scale samples from some model. And I'm denoting that as Q, but we could just think of that model as the geometric mean plus throw some error on it, right? It could be anything you want it to be. But in doing so, we're going to have more robust inference, right? We're encoding rather than placing our beliefs on the scale of the system into some weird assumption that's made by the normalization, we're actually explicitly saying this is how we believe that the scales of the system behave. So the scale models can be based off anything, and it could include normalizations. It could include biological knowledge. Maybe we have reason to believe that the antibiotic that we gave in our example decreases microbes by 10%. We could use that and incorporate that knowledge in. Maybe we have some estimate of flow cytometry that can help guide us how the scales differ. So we're just going to then show how we can use this information within ALDEx2 rather than relying on the normalization. And in doing so, we can kind of greatly improve our inferential results. All right, so I know that was a lot of information to throw at you guys. We're going to get started with the coding next. Does anybody have any questions on what I'm even talking about, where I'm going, anything at all? Yeah.
I don't understand what ALDEx2 is used for.
Oh, differential abundance and differential expression. Sorry.
Okay. So it's specifically for like RNA-seq?
That's what it was developed for. People use it for all sorts of things anymore. Sorry, I went back too far. Yeah, anything differential abundance, differential expression. You can use it to fit linear models too. That just changes like this step right here. It's still the same. You use Dirichlet samples, you transform it, and then you fit the model on the samples, and then you aggregate over like your betas over your samples, if that makes sense. Yeah. But yeah, anything along differential abundance, differential expression, any type of linear model you want to fit, basically, is what ALDEx2 is used. Sorry, I didn't touch on that.
No, you're fine. I was like super lost. Like, this is great, but what do I do?
Yeah, well, and I've been working on this for so long, so it's like easy to take for granted, right, what you know. Yeah. Any other questions?
So you were saying that we introduced this new assumption and we use out of some kind of prior knowledge.
Yeah.
Isn't it risky that we introduce intentionally like bias? Like we expect something and we put it there and then we get the result that we expected because.
So I would argue that using the CLR is almost more risky, right? Because you don't even know what it's saying. Right, if you want to do differential abundance or differential expression, you can't solve that problem fundamentally without knowing scale of the system, right? And it is like, if you apply DE-seq2, what is DE-seq2 saying about your scale, right? Do you know? I mean, I don't know. Like, that's the problem with these methods is that it's often hard to interpret what's going on. We're arguing that instead of just relying on these normalizations to bridge that gap for us, let's just be explicit and say that this is the scale model we're assuming, and our results hold under the assumption that there's a 10% difference plus minus some error bound, right, between the conditions. We fully acknowledge that people can misuse the tool, but people can misuse lots of tools, right? Yeah, does that answer your question?
Yeah, I guess, can you, maybe it makes more sense when we do the coding, but like the scale itself, so like, are you like assuming the scale is like at 1,000 microbes or whatever it is, or?
So it would depend on the problem, right? The scale is dependent on what the question you're interested in. If I'm interested in knowing how the scale changes pre and post antibiotic administration, what I'm trying to get at is the total number of microbes in your gut. It'll turn out that I'll show it doesn't matter the number of microbes in your gut, it just matters relative to the next person, right? And so what we're saying is you can encode the knowledge, not necessarily like there's 1,000 microbes in the gut,
Okay.
just that condition 1 to condition 2 might have this kind of change. Or that we have these outside measurements that are informative of the question we want to ask, and they're pointing at this kind of change, and we'll use that. Does that make sense?
Yeah, because I was thinking like the scale will like presumably change, right? Especially with antibiotics, right? Like one system will be much lower in number than the other one. So is it like, is the scale like accounting for that, or is it assuming that both should be at a
So the
certain number?
scale, so the beautiful part about scale models is yes, we can account for that. A
Okay.
scale model can be anything you want it to be.
Okay.
The whole point is that you're explicitly saying this is the scale. This is how I think the scale of the system behaves. Recall ALDEx2 previously made this assumption that the scale is the geometric mean. Which, think about our antibiotic assumption. That doesn't make sense. And actually, it's like a total aside. That's what's going on in this plot. The geometric mean is saying there's more microbes in your gut after taking an antibiotic than not, and which is what's causing this unacknowledged bias. We're just saying rather than rely on whatever implicit assumption there is, let's make it an explicit part of the modeling process.
And so then that also like translates over to like, let's say like when you use like different sequencing platforms, right? So like the sequencing depth, right? It's gonna change. So it's also, inherent for that. Okay.
It's whatever you can use whatever information you think is informative for the question you're trying to answer. You could also even like, some people have talked about like, if you have like housekeeping gene type experiments, you could use that information to help build your scale model. If you know certain things aren't supposed to be changing, you can use that as a benchmark. We're just, again, it's this argument of rather than relying on some intrinsic definition imposed by a normalization, whether it's ALDEx2, DEC2, EDGAR, whatever it is, we're arguing that step away from that, let's be explicit and include scale models, because to do this type of inference, you have to make a statement about scale. So let's just put it in the forefront and not use whatever's just implied. Any other questions? Are we all good? Okay, so now we're actually going to get into the coding part. So for anybody who's used ALDEx2 before, the change that we made is actually really minimal. We only added one additional argument. And I'm going to show how we've done this all through a simple simulation example. So the first data script's going to be needed for this, one simulation R file. And so to show for the rest of to show the capabilities for the rest of the section, I'm going to be using this antibiotic example still. And basically I simulated this data. We have a precondition and a post-condition. I used Poisson sampling to sample the true abundances. This is what we have. You'll note that there's a couple of taxa that are changing, like this one right here, there's one right there, and then there's two down there. You also notice that it's always decreasing, right, in the post condition over the pre, it's lower. Then I use this data set, and I use multinomial resampling to kind of mimic the lack of information that happens when you go through your sequencing steps and your sampling steps and all of that good stuff. So all of the simulation functions for making this data is available on GitHub, but we're not going to run it now just for brevity. But if you want to see how I did it, it's there. All right, so the first thing to do then is to read in this data. ALDEx2 is the main package you need. The rest of it's just because I coded in tidyverse. RDAT right here stands for your resampled data. So it's what after the multinomial sampling, we don't have any information of scale in the data. And the flow data file we use later, that's just some mock flow cytometry data that I created, which we'll discuss more later. Right, is everybody good?
Sorry, Michelle, I'm confused. This is on your GitHub page.
It's on my GitHub.
Can you? Yeah. The link.
It's all available in the GitHub. If you open up the R project, everything should just be like set for you in terms of file directory and structure. And then all of the scripts that I'm talking about are in the scripts folder. Here's all the code that I'm about to walk through.
I'm so sorry, could you see?
What do you need to?
I need to be able to see like the actual like site name or like the name of your. Oh, is this the dog workshop one?
Yeah.
Oh, I don't, sorry, it's like I don't have access.
You don't have access. Oh, this is on my GitHub.
Oh, it is.
Yeah. Sorry, I didn't blow up the address at all. It's just github.com/Michelle Pisner/the dog workshop. Just like that is. Should be public. Anybody should be able to get access to it. Anybody need more time to get this stuff? We're all good. Okay. All right, so once you have the data read in, just a quick look at the data just to kind of get you guys oriented to what I made. Y is like a ASV OTU table. We have our 20 taxa here. Remember, 4 are changing, 16 aren't. And we have all of our samples. I think I did 50 samples in each condition, which is big, but if you want to play around with the simulation, you can reduce the number of samples there. And then you can't see because Zoom's cutting it off. We have this pre and post condition vector. That's what we're testing our differential abundance on. So we're going to feed this in ALDEx2. Goal is to try to understand which taxa change between the pre and post. So then the first thing we can do is fit the regular ALDEx2 model. So the way that you do that is pretty simple. This is just using all of his standard stuff. You just call the ALDEx function. You feed in your count table and you feed in your condition vector. And that's it. Now, if you want to change like how you're testing things and some of that stuff, we can talk about that. But by and large, that's all you need to do to run ALDEx2. So this is also implicitly assuming that we're drawing 128 Monte Carlo samples, which you can change with the Monte Carlo MC.samples argument. But let's just take a look at the output real quick. Okay, so you'll note that we have a row for every taxa. This RAB all, so Greg picks the most informative names ever, so they're not very informative. RAB all basically is just the CLR coordinates if you would take the median across all conditions. So you're blind to the pre and post status. The next two columns, the RAB-win pre, RAB-win post, is just the median CLR if you restrict yourself to the precondition and the post condition, like respectively. One's looking at just post, one's looking at just the pre. The difference between is just the difference between those last two columns. The difference within is an effect of the noise within each group, within the post and the pre. Again, all of these are like median values. The effect size is basically taken from dividing difference between divided by the difference within. So how much we see between our groups versus how much is just like the base level noise. Overlap is a notion of how much of the effect size like crosses zero. So like, is it centered about zero versus not? And then really the like core thing that people would use to determine differential abundance is this WE-EP column, WE-EVH, WI-EP, and WI-EVH. What they are is just two different types of tests. WE.EP is the p-value that corresponds to just fitting a regular t-test. The WE.BH is the Benjamini-Hochberg corrected p-values from that t-test. And then WI-EP and WI-EVH comes from using a Wilcoxon non-parametric test at the same time. So this is the standard output that you get from ALDEx2. So if you wanted to know what was significant, you'd probably filter on either WE-EVH or WI-EVH and see what's less than whatever your favorite threshold is. Does that all make sense to everyone? Okay. So the practical implication for this example, right, is that we can recreate the fact that we're getting everything as significant. So this all tags are returned. If I filter again, WE-EVH is the Benjamini-Hochberg on just the regular t-test, less than 0.05, getting everything back. We know that's not true, right? We know that only 4 are true, so super high false discovery rate because 16 things are false out of 20. All right, so that is the existing ALDEx2. So if you've used it before, this is probably the interface that you're used to. Nothing has changed from that regard unless you start playing around with the parameter that we've added called gamma. And gamma is written into both the ALDEx function before, which is an ALDEx CLR if you've used it. If not, don't really need to know that. But gamma can either be a single numeric value or a matrix. If it's a single numeric value, it controls what we've built in as a default scale model. If it's a matrix, that's how you could do some of the more like advanced things we talked about, where like, suppose you have flow cytometry data and you want to build scale samples off of that, or suppose that you have some outside information that you know the scale between conditions is changing in a certain way. That's how you could still input that information through gamma. We built in this default scale model mostly because we find it'll be helpful in practical implications, just as a way to get researchers using the tool. So we're basing it on errors in the CLR normalization. So recall before, ALDEx2 is just assuming everything, like if we get rid of this term right here, ALDEx2 is just assuming this log W perp is equal to the negative mean of the log of the composition. Now we're just adding noise onto that in a pretty special way. We'll decode what that means in a second, but I just want you guys to like draw your attention to the fact that there's this gamma term right here. That's what we mean when we say like you input a gamma, you're inputting some level of noise right into that assumption. So let's take a step and try to decode this. So this up here is just the equation that I presented on the last slide. All right, so we talked about before how the geometric mean applies a point. So that's what I'm showing here in the pre and the post density spots. It's just saying with certainty, you know, the scale is -5 or -5.15. This lambda xn term is adding noise, and you'll notice we're only adding the noise to one condition, and it turns out we can do that and it doesn't really matter. And then the final result we get right for the log scale samples is just adding the two together. So you'll notice for the precondition, we're just it's the CLR. For the post condition, we have the CLR now with some uncertainty added to it. And the reason we only need to add uncertainty to one condition is because it's not actually the scale of W perp that you care about when you're doing most of your analysis. It's like the difference between the two groups, if that makes sense. So what we care about actually is this estimate of the difference of scales. In the CLR normalization, it would just be subtracting two numbers. You subtract 1 minus 2, you just get it right, another number back. That's what's happening here. Whereas in the default scale model, we have that uncertainty built in. So now we're getting a model that's based off our normalization value. You can see it's kind of roughly centered, but still we have some uncertainty that's dictated by whatever value of gamma I chose. And so basically what we're saying is that 95% with our default model, you're just adding that like 95% of your samples fall within this plus minus 2 gamma of whatever you would have assumed under a CLR assumption. Now, I know that's like a lot to try to wrap your head around. So does anybody have questions? So the biggest thing to remember is that gamma dictates how much noise you're adding to the assumption that's already implied by the CLR. And so we've coded it very easily in ALDEx2. All you add is that gamma equals argument. And so we added gamma equals 1e to the -3. From everything we've seen in the previous slide, this is just going to be what would have been returned anyway, right? We're adding 0 noise, the CLR assumption essentially, or very little, right? Standard deviation of 1e to the -3, which has the implication that we can recreate what was implied by the CLR with the scale model. This plot right here is the plot of effect sizes with the original ALDEx2 effect on the X and the CLR scale on the Y. And basically we can recreate by adding no scale noise.
How are you actually choosing the number of gamma?
How do you choose gamma?
Yeah, like what made, like, how do you decide that it needs to be 1 to the -3? Hey, there it is. Okay, sorry.
Yeah, we recommend just a value of 0.5 in general, just because it corresponds to this assumption of half or double the normalized amount. Although that's not like a hard, fast rule, obviously. But we also argue that any value of gamma is going to do better than what ALDEx2 would have returned because you're adding uncertainty to the CLR, which means you're going to reduce your number of false positives. Even if gamma is 0.1, you're going to reduce your false positives over if you were to just use ALDEx2. If you set gamma something super high, like gamma is 20, you're going to get probably nothing back because you're basically saying we don't know anything about the scale, but you still then you're like reducing your false positives at the rate of not discovering anything.
Okay.
So adding in gamma, even just a small amount, 0.25, we washed away all of our false positives essentially, have 3 taxa that are returned as significant, which is a pretty remarkable improvement just for adding a little uncertainty in the CLR assumption. If we set gamma equal to 1, we've lost everything. Because it's basically corresponding to we just don't know what the scale is. And if we don't know what the scale is, we don't know what's changing. So there is definitely a balance between like noise added, the amount of false positives, false negatives, all of that good stuff. Sure.
So let's say like transitioning from like studying the gut to the mouth, right? And the scale of the mouth is usually a lot smaller, right? There's less microbes in the mouth than in the gut.
Mm-hmm.
How would I play around with gamma in that scenario? Like, should I even play around with gamma in that scenario?
So in that scenario, you can. We talk about it. So we're going to have a paper coming out pretty soon, and we talk an example about that. Like it's a little trickier to do because you have to play around with what's implied by the CLR, but then you can back out a value of gamma that way.
Right.
But for something like that, we would argue even just adding some scale uncertainty is going to assume that your results are a little bit more robust, whatever assumption you're making.
Okay.
If that makes sense.
And that's just like transitioning to any ecosystem. Like, I mean, like even like the soil or anything like, so I was just thinking like, in my idea, I would think the mouth has less noise than the gut just because it has a lot less, but that may be a wrong assumption.
Well, it might, and that's very well true, but this is all coming back to like, what's the plausibility of assuming that the scale of the microbes in your gut is related to the geometric mean? Right.
So it's kind of dependent then.
Yeah.
With like of the biological knowledge that we.
Yeah, it is dependent on some of the stuff, but I think you have to remember that the CLR that's used in ALDEx2 is not because we think that there's biological reasoning behind it. It's because it's mathematically, like that's why it was chosen. It's mathematically convenient. And mathematically convenient does not necessarily equal good, robust stuff. So this is where like getting into like instead of dealing with our errors and normalization approach, which we think will be relatively reasonable for most applications, where you can just create your own scale model. And so this is like a back end where gamma could be a matrix, and you can pass any matrix you want to it, as long as the dimension of the matrix is n, which is your number of samples, times s, which is your number of Monte Carlo replicates from that first step with Dirichlet sampling. And also the other like important little piece of it is that they're what you believe are samples of the scale, not the log scale. ALDEx2, the way it's coded, will just kind of handle those transformations itself, but you just need to make sure that they're samples of the scale. So this is like going back to the one example I think that Sterling kind of mentioned, that like if you believe that the microbes between conditions decreases by a certain percentage, you can encode that in a model. So here basically I'm saying that we believe that on the log scale there's a 10% decrease between pre and post. This can be adapted using this ALDEx.MakeScale matrix function to draw samples where we're encoding that mean difference. So what we're saying here with this line of code is basically make me a scale model where the difference between, where we have a value of gamma, so a value of noise of 1, but we expect that the difference between the pre and post condition, this is only assuming 2 samples, is 1 and 0.9, so a 10% decrease. And we want it to be on the scale of W, not the log scale of W. And so we can use this to make matrices, like matrices that we can pass to gamma to encode our scale beliefs. So how do we code that then in ALDEx2? Back to our example, we have scales, which is the mean assumption that we're assuming between the precondition and the post condition. Then we use this make scale matrix, which basically is saying assume a noise of 0.15, use the means above, so we have a 10% decrease on the log scale between the pre and the post, our condition vector, which tells it what is actually in the pre and the post, which samples, and return them on the scale of W, not the log scale. Feed that into ALDEx2 using the same argument, and we're getting perfect inference now. Everything that's being returned is a true positive at the expense of no false positives. This is mostly because, right, it's a simulation and the scale model is reflective of actually what the truth is.
And this would have been, this is from flow, like simulated flow cytometry data.
So this OTU is simulated just using like Poisson. The way that I simulated this was I used Poisson sampling to simulate like W, so stuff that's on like that actually has the abundance information. And then I use multinomial resampling to subsample. That's how I created Y. So I have like loss of sequencing information reflected, but then I can create the flow cytometry data fake because I have access to what the true counts are. And I use that information to make some noisy flow data.
Okay.
Does that make sense?
Yeah, so if you were running this in real life, you
Yeah.
could use flow cytometry on bacterial cells to get an absolute count of the cells in the sample.
Yep. And if that is reflective of what question you're asking, then you absolutely can use it.
Okay.
Right.
Could you use qPCR for 16S?
If it's reflective of what question you're asking. Justin will tell you, and you know, Justin, that there's all sorts of potential, like there's subtle mismatches maybe between some of these outside measurements and questions you're asking.
Yeah.
I would argue it's probably a lot better than just saying negative mean log geometry, right?
Okay.
But again, you still have to think about like, and what it is, what I'm feeding in reflective of what I'm trying, like the scale of my question that I'm asking.
Okay.
Right. Scale doesn't need to be absolute abundance, which is something that I didn't measure, right? Scale, it does depend on your question. Our research question here is which tax would change in absolute abundance, so it's obvious. What if it was relative to the length of the colon? Now your scale is some measure that's not necessarily absolute, but there's also a piece of missing information. So this right here, I think, goes to Emily's question of how do you use scale if you have outside measurements? So again, can be used if they're informative of whatever question you're asking. Here we're going to show how we basically, Z is my, like, it's a flow cytometry measurement. We're going to show how you can just use, like, if you have that collected, you can build a scale model based off of that. And so here's the fake flow data I created. I created 3 samples for each sample in my data set. This is just some code to collapse it down. So I like over my replicates, I'm just taking the mean and the standard deviation. Once I have the mean and the standard deviation in the top code, I can use it to sample for each sample some values of scale based off the flow cytometry data. And doing that, feeding that into gamma, again, gives us perfect inference. So gamma is super flexible. Errors in normalization, which we would argue should just become the default. Using the CLR is mathematically convenient. Results really hinge on that statement, so why not add a little bit of error to it? But if you do have access to outside measurements, if you do have strong beliefs about whatever system you're studying, this can be encoded this way too. All right, so just to recap and compare across all of our methods, here are our 20 taxa again. Here are the results that we had for everything. Original ALDEx2 and what we created with the default scale model, remember, returned everything, which was 16 out of 20 taxa were false positives. Adding just a little bit of noise, we dropped down to 3 of 4 of our taxa that are actually significant or significant. Everything else is gone, which is a massive improvement. Setting gamma equal to 1 basically wipes everything out because we're making a statement we don't know anything about scale. So how can we say if things are changing? The knowledge-based model, which was that 10% decrease, we got perfect inference. The flow-based model, again, we got perfect inference. Obviously, this is a simulation to show you how things work, but it can lead to some pretty drastic improvements over just using a normalization model. And so there's one more thing to talk about that we added, and then we can work through the CellX example together. So we added a sensitivity analysis. So this whole time, right, there has been some just like, how do you choose gamma? Why do we set gamma as 0.5? Well, we're arguing, why do we even need to set what gamma is, right? We can use our default scale model and test over a wide range of errors of different values of noise. And so I've coded in a couple of functions to make it pretty easy to do, at least I hope it's easy to do. Basically, you need 2 inputs. One is an ALDEx CLR object, which you can create out of ALDEx2 right now. Another is a vector of the values of gamma you want to test. So this is how much noise again. And then you run a sensitivity analysis function, and then you plot it. So it's running these 2 additional functions, and it will basically rerun ALDEx2 over all of the values of gamma you want to specify, and you can see which taxa are significant when more uncertainty is added in your normalization. We think this will be pretty powerful. We also think it would lead to more reproducible inference, because you basically can say, we found that this taxa was significant up to a gamma, like up to a certain amount of scale uncertainty. And so it allows people to decide whether or not they believe whatever scale you're assuming is good or not. So again, running it's pretty simple. Vector that you want to test over, create your CLR object, run these 2 functions that are new to ALDEx2. And what you get out are a couple of plots. This one shows the dropout rate. So as a function of gamma, the number of significant entities just drops off somewhere between about gamma of 0.25, 0.5. And this plot, I think, is a little bit more informative. Shows you which taxa are significant as a function of gamma. So we can make a statement like taxa 20 is significant because it's colored, mine is colored, up until a gamma of about 1. And so if you were reporting findings on it, you could say, up until the scale model with this much noise, we find that this taxa is significant. 14 and 15, which are our other true positives, drop out somewhere around like 0.25. And so it just kind of gives you a way to assess how things change.
Maybe silly question. So is there like a lower gamma level is like better, or
More noise would actually be better. Okay, so the way to think about it, it's a good question. The more noise you're adding means you're adding more uncertainty. which means your result is holding as significant under even more random scales, right? Think about it like we don't, I guess a concrete way to say, step away from the normalizations. We don't know what the scales are actually doing. It'd be a stronger statement to say this tax is significant if it's 10% decrease. It's also significant if it's 15. It's also significant if it's 5, right? It's significant over this whole range rather than it's just significant at 10%. And that's kind of how you can think about it here. The higher value of gamma is just saying we would find that significant under a wider range of possible scale values than if gamma was smaller.
Yeah.
Any other questions? Okay. So then the other kind of example that we're going to go through, and I can like code it with you, is the Celix example, which is native to ALDEx2. If you've looked at any of the vignettes, this is the data set that Greg Lohr uses and plays around with. And so basically a very short description, Celix stands for Selected Growth Experiment. The way it was conducted is there's these 1,600 different gene variants. The whole goal is to understand which ones confer this growth phenotype. It was conducted in such a way that if they added a bacteriostatic toxin, certain things would grow, but other things wouldn't grow. And so we have this one-sided asymmetric kind of growth issue. They have two conditions, a selected condition where we allow for growth in some cell lines and a non-selected condition where nothing can grow. So, right, we really have this asymmetric growth is only allowed on one side. Scale issue. Here's the citations where the paper was originally discussed and originally published, but it's a short, simple 7 conditions in each group, 1,600 variants, just non-selected, selected type thing. So the code for it is in the Celix analysis, and like I said, the data set is all just built into ALDEx2. We have our 1,600 growth variants split across the 14 conditions, non-selected and selected. Just taking a look at what we have between the different groups. The data looks quite different between the selected and the non-selected group. The reason why I like this data set, because we also have a notion of which is true. So we do have some ground truth. There's only 27 phenotypes that have been shown to be like biologically active. So just fitting ALDEx2 with no other modifications takes a little bit because it's just big.
Can I ask a question?
Yeah.
That's running. So for like the cons variable.
Yeah.
So like when you're working with other data sets, like if you're not like, is that just like specifying how many replicates that you have of, or like how many samples you have of the NS and the S?
So this is just like a condition vector saying that sample 1 is from NS, sample 2 is from NS, sample 3. If you're working with more complex data sets, you can feed it in a matrix. That's kind of, which would again just specify like your covariate data for each observation. And there's some talk, like if you look here, you can feed it in a data frame. Yeah, so it's like your covariate information. All right, so filtering it down on what's significant using the t-test, Benjamini Hochberg, we're getting 50 false positives, 18 true positives. That's just comparing it back to the truth. Now, if we add scale, gamma of 0.5, this is our heuristic value. Basically what we're going to see is the dropout on the false positives. I think with 1 true positive going away, which is what we would expect to happen. And so this is really part of our argument why this is just almost like a no-brainer to do if you're using ALDEx2, is to incorporate some scale uncertainty to see which results are really sensitive to that CLR assumption. So we lost a handful of true positives at the of true positives at the expense of quite a bit false positives. Then I have some functions basically just to test over different values of gamma and to plot them. So here, right, we're just testing this vector. It's basically a sensitivity analysis, testing this vector, running ALDEx2 with our different values of gamma, filtering down, calculating the number of true positives, calculating the number of false positives based off the ground truth, and plotting it all up rather than running it. Basically what we're seeing is that there's a huge dropout on the false positives. Obviously a steady decline coming with the true positives as well. But especially for the smaller levels of gamma, it's basically you have nothing to lose by not incorporating some scale uncertainty in this example. We'd also argue that like something like this could be useful depending on what your goal of your experiment is. If your goal is just to like identify potential variants that you would want to study more and want to spend your research dollars in, obviously the best place to start is probably those that are still significant when there's super high amounts of scale uncertainty added. As like candidates. And again, we can look at the same thing with the sensitivity analyses using the same kind of ALDEx CLR, run the analysis, plot it, and it's going to give us a very similar flavor to what I just showed. We do have some significant taxa that are true positives that are significant even when a huge amount of scale uncertainty is added. Most of the other ones, especially the ones that were false positives, drop out pretty quickly with just small amounts of noise, which again, I think shows that it's just silly not to use it if you're using them. And so that is the materials that I have prepared. I'd be happy to answer any questions, help people troubleshoot with their own data sets, talk about anything more. Yeah.
So have you like done like maybe like a meta-analysis where like, or has someone else like taking different data sets from different, let's say people looking at the gut microbiome and then using this tool and then get more consistent results? Or is that something in the works?
So this paper here, the Glor, Nixon, and Silverman, is on bioRxiv right now. That has 2 more examples of scale. One using a transcriptome data set, one using like a vaginal microbiome, where he showed that if you use the housekeeping information, you can increase the number of discoveries. The Beyond Normalizations paper that's forthcoming, we have a couple more examples in it too. So, yeah.
I have a question, but it's about like my data, so I don't know if everyone wants to listen or not.
Yeah, I'll be willing to help you.
Does anyone have questions before I ask?
So this is the new functionality of ALDEx2. So if you download it off Bioconductor, there will be a gamma parameter at the very least. Now you know what gamma is. I would argue that it's silly not to use it, even just to see how errors in the CLR normalization are affecting your results, right? If you have something where if you add just a tiny bit of uncertainty in the CLR and it goes away, I wouldn't hang my hat on that in a paper.
Does this presentation with the references exist anywhere?
It is on my
your
GitHub.
It's on your GitHub?
Yeah.
Where
It's in the slides folder.
is it?
Yeah, it's the HTML. It's
Yeah,
not, it was
there
built in
is-
our markdown.
Maybe it's it says like, I'm just getting like, sorry, but we can't show you these files.
Yeah, you have to download it.
It's
You can't
probably
open
because it's
HTML and GitHub.
large. But the HTML is right
Oh,
here.
And then okay.
Download it.
Yeah, they GitHub will let you fix files and stuff.
Oh, okay. It's just, yeah, it wasn't even showing like. I get it.
Yeah.
Okay. Thank you.
Welcome. Any other questions? Right, I'm happy to answer like specific data questions or if anybody has ALDEx2 questions. Got way too familiar with the package.
I have a question because I'm doing a study and I'm trying to apply like some more like complicated GLM model.
Okay.
And kind of like the write-up at the end is really short, so I've been having a little bit of trouble. I think I got it to like run correctly, but I'm just confused about the results.
About like GLM.
Yeah, okay. Because, yeah, so essentially I have like males and females, and then within that I have wild type and mutant, and then under each of those I have control and treatment.
Okay.
So I think I like properly accounted for all of that, but then, yeah, the output just seems to have like results for each, like a couple comparisons, but not like considering all of them.
I see it real quick.
Yeah. So, yeah, so this is my output. I can also show you the I set it up.
Okay. Yeah, show me set up really quick. So the output for the GLM function. That's the effect size. The ALDEx GLM will be more like what you're used to interpreting, like with betas and stuff like that,
Okay,
and interpret it for every thing.
okay, so
But
the
it's-
GLM test is actually one.
Looking for like beta, like you have your taxa and you want to know how like sex and I forget what else you said changes within each taxa. Yeah, you would just want to take a look at the ALDEx GLM. The whole argument with scale and all of that stuff can still be applied to the GLM function.
Okay,
Default scale model should work there.
I think it's mad because I updated.
Yeah, but like, so I have it running on the simple example right here. So this is like, now we're testing this model matrix. This is Greg Lord's example, where we have these two binary AB with an intercept. And the GLM test now will give us basically an estimate for the intercept that's aggregated over the Monte Carlo replicates. So this is over the S replicates, an estimate of the standard error aggregate over the replicates, the t-value, and then it does it for everything. So you can use it like this is the estimated effect of A on this first.
Okay.
Yeah, and then the significance is all similar, aggregated. You have both p-values that are aggregated over the replicates, but also adjusted p-values.
I think that makes sense. So like the A p-value adjusted would be like the p-value, but just in reference to that.
It's the test of his beta. Right, so like the model we're fitting is right, like y equals beta naught our intercept plus beta 1 times A, indicator of A, plus beta 2, right? It's the test of his beta 1 equal to 0 or not. And so it's saying for A, basically A is never significant. Whereas for B, like for example, let's see if any of them are significant. Here, right here. This would say that there is an effect of B on this specific variant holding all else constant, just like you interpret regular linear models.
Go ahead, Sterling.
So the example he gave was like two conditions, right? Like pre and post, like
Yep.
it's a categorical.
Yep.
Are you able to run this with continuous variables too?
Yep. So ALDEx2 is written in a very modular way. ALDEx, the function I was showing, is actually a wrapper to like this ALDEx CLR, ALDEx T attest, ALDEx GLM. You can use the GLM module to specify continuous covariance. So like I could have alternatively said here that A is a normal random variable, right? I need 14 of them though. And that would be a legit model to run.
And so like, let's say you just, yeah, like a range of BMI, you just put like the BMI range in there then?
Yep, you could put, it's flexible, it will do any type of linear models. But the results will look relatively identical. And again, you can still add scale into that. Now we just have an estimate for A, that's just that continuous covariate that was garbage when they made up.
Michelle, my question was, like, this is really cool and exciting. I can imagine, like, for example, in Erica's lab, we're often submitting papers to like the Journal of Animal Science, where reviewers and the audience are gonna be hardly informed on anything bioinformatics. And if we were to say in the methods, like, oh, we used this function to make an inference that's scale sensitive, reviewers and the audience would have no idea what that is. So can you suggest like wording or a way to kind of like word it for more of a lay person?
So the fortunate part, I feel like on that regard now, is that this is all documented in ALDEx2 on Bioconductor, right? And so the citations there, the papers that Greg have done, I think help with that a little. It's not like I just
Yeah.
made this package up and pushed it to GitHub.
Sure, yeah.
But yeah, I think the way to talk about it would be, we considered potential errors in the CLR normalization by adding some scale uncertainty. And we found that these results held even under whatever potential error we considered. I think it could just be something as simple as that.
Okay.
I think this question was already asked. I'm just like,
You're fine.
anyways, so this makes sense when you're comparing like A versus B, like taking antibiotics, not taking antibiotics. Does this work for systems where you have like multiple treatments?
Yes.
Okay.
So it does work for those systems. You could still use the same ideas to create scale. The default scale model changes slightly, mostly because we just add more uncertainty where the more covariates you add, because we find to believe, like, right, if this pre and post conditional affects scale, maybe it's also that, you know, male versus female will affect scale too. So we had just a little bit more uncertainty, which changes interpretation slightly, but you can also still put in whatever you want through this matrix approach too. So like if you were doing a system with all these variables and still had access to flow cytometry, as long as it was helpful on the scale that you're interested in, there's no reason that you couldn't incorporate that information.
Are there too many, just playing off of that, variables that are like, it's just too much in the model, it's just like way too much uncertainty and then blows up, you know, like it won't work. Yeah, so what's the threshold there?
In terms of,
Or just number of like.
oh, I mean, you always
covariates?
want to use good statistical practice, right, when you're building models. So it's a balance of not overfitting your data.
Right.
I would just go back, because like you have to remember, like ALDEx GLM is just fitting a linear model. I mean, just as like, there's obviously some intricacies, but just all of the principles that you've learned from Stat 200 on best practices, or, you know, whatever grad courses you've taken about fitting linear models in a reliable way, still kind of hold here.
That's helpful, thank you.
Yeah, so like, yeah, it doesn't go out the window.
Sorry to keep asking questions, but like, have you also like compared the results with other differential abundance tests? Like is it, I like the, I mean, the other differential abundance tests also use GLMs, right? But like,
Yeah.
is
Are you
this
talking
is
like,
unique and that it includes like the uncertainty?
Are you
Like, so you're
talking like
getting
if
more
we could-
false positives or false, like a more or less, like,
are you talking like, have we compared it to like DSeq2? Or are you talking like,
so I only like, I use Maslin 2 for a lot of my differential abundance
Okay.
analysis. I've also used like Ancom BC in the
Okay.
past.
so basically every method makes an assumption about scale. ANCOM makes this weird sparsity assumption that like, well, it's not sparsity, it's like that most two are changing or something, like no more than two are changing, but every method makes an assumption on scale. You have to, right? We have no information about it or very little information in the collected data. So we need to bridge that gap somehow. So this problem applies much more wildly than just ALDEx2. It just so happens that ALDEx2 has a very super intuitive form that makes it super easy to open up, manipulate, and get to incorporate scale uncertainty. But yes, other methods, DSeq2 still suffers from this. In the one paper, we're going to have a benchmarking study that compares a bunch of these to basically show that it affects anything that makes a normalization is making a scale assumption about scale.
Well, yeah, but like for my question is that, so are you getting less taxa with this than like before, like less false positives?
That's generally the idea, right? You'll like, if you add uncertainty, you're always going to get, if you add uncertainty via the default scale model, you're always going to get less false positives than if you would not add uncertainty, right? Just the way it's constructed, since it's based off the normalization. If you're using your own scale model, you're gonna get results that better reflect the biology, right? If you believe that this holds, your scale model encapsulates behavior, then your results will hold under the assumption you encoded in the scale model.
Okay.
So like if you were truly, you know, if you believe the flow cytometry is reflective, incorporate that information, then you're getting results that are consistent, that hold, assuming your flow cytometry is consistent with what you believe, or with whatever notion of scale. Does that make sense?
Yeah, so like, it seems like this is more confirmatory than like, if you're like maybe in the more like exploratory part of your projects then, or?
Well, it all depends on what you want to do, right? You could even use something like this if you are in the exploratory phase, but you have limited dollars to spend, right? And you want to know what is most likely true signal versus false. You could do something like the sensitivity plots that I showed, right? And say, oh, hey, these couple of tacks are holding up even when I throw insane amounts of scale uncertainty, let's go after them first. In general, I would say this is definitely like a confirmatory, yes, we believe that these results are robust to scale though. I think it can kind of be used both ways, if that makes sense.
Yeah, that's why I was just like curious, like maybe like I'll use another program if I want to like, if I don't want any false negatives, right? Because then they'll pop up. But then if I want to confirm something, I'll use this to
Yeah.
kind of narrow it down.
It's just the main story is just like, if you take away nothing else from this talk, it's just normalizations imply something when you're doing these methods. Every method has to make one and make some assumption. Whether or not it holds for your data set is maybe, so just think about it, is like the argument. And furthermore, like, it's hard for, I'm a statistician, I can't interpret some of the like supplemental information that's put out there. It's just, it's hard for these things to actually understand what's going on in these methods. And so like transparency kind of is key. Yeah. Any other questions? Okay, I'm happy to stay back if anybody has questions like more specific to their stuff. Yeah, thank you guys.